Welcome friends to a new session on genetics. In this session, we will discuss the analysis of mutations in biochemical pathways and one gene, one enzyme hypothesis. Microbial genetics is concerned with the transmission of hereditary characters in microorganisms. Microbial genetics has played an important role in developing the fields of cell and molecular biology and also has found applications in agriculture, medicine, food and also pharmaceutical industries. In microorganisms, the hereditary is analogous to those in multicellular organisms. In both eukaryotic and prokaryotic microbes, the genetic material is DNA. RNA viruses are the only known exceptions to this rule. The heritable changes in the DNA, that is, mutations occur spontaneously and mutagenic agents increase the rate of mutation. The susceptibility of bacteria to mutagenic agents has been used to identify potentially hazardous chemicals in the environment. For instance, the Ames test was developed to evaluate the mutagenicity of a chemical as per the following. The Petri plates containing a medium lacking in, for instance, the nutrient histidine is inoculated with a histidine requiring strain of the bacterium Salmonella Typhimurium. Thus, only cells that revert back to the wild type can grow on the medium. The increase in the number of mutants compared with unexposed plates can be observed if the Petri plates are exposed to a mutagenic agent, and a large number of revertants would indicate a strong mutagenic agent. A revertant is a mutant that has reverted to its former genotype or to the original phenotype by means of a suppressor mutation or else by compensatory mutation somewhere in the gene. For such studies, microorganisms offer the advantage that they have short mean generation times, are easily cultured in a small space under controlled conditions and have a relatively uncomplicated structure. The reactions involved in the biosynthesis of a compound occur in a precise sequence so that the product of previous reactions serves as substrate for the next reaction. In a biosynthetic pathway, if a particular reaction is blocked, the substrate for that reaction will accumulate, while the product of that reaction and those of all the subsequent reactions will be absent. A biosynthetic pathway can be worked out using biochemical mutations, since mutation in a particular gene leads to block in a particular step of biosynthetic pathway can be explained easily by an example of eye pigment in Drosophila. Let us see about eye transplantation in Drosophila. Before having initiated his work on Neurospora crassa, George Wells Beadle, an American scientist in the field of genetics and Nobel Prize winner in physiology or medicine, had conducted eye transplantation experiments in Drosophila in collaboration with Boris Ufrusy in Europe. Beadle and Ufrusy transplanted larval embryonic eye primordia from one mutant type into another and studied the development of pigment in such transplanted eyes. The results of these transplantation experiments are presented in this figure as well as in the table to be followed. This figure shows eye transplantation experiments in Drosophila showing autonomous and non-autonomous development of implanted embryonic eye discs into various phenotypes. The table shows color of eye primordia developing in hosts of different mutant types in Drosophila. It can be noticed from the table that when wild type eyes are transplanted on a vermilion or cinnabar host, wild type eyes develop, indicating that phenotype of eyes in this case is independent of host. In reciprocal transplantations, when vermilion or cinnabar eyes are transplanted on wild type, these transplanted eyes also develop into wild type because a diffusible substance, probably an enzyme, from host helps the transplanted eyes to develop into wild type. Crucial results were obtained when reciprocal transplantations were made between vermilion and cinnabar. When vermilion eye is transplanted on cinnabar, a wild type eye develops, indicating that a diffusible substance from cinnabar enables the transplanted vermilion eye to develop into wild type. However, when cinnabar eye is transplanted on vermilion, no diffusible substance is available which would enable cinnabar to develop into wild type. This indicated that vermilion color appears due to a block appearing earlier in biosynthetic pathway than in case of cinnabar. The brown pigment in the eye of Drosophila called umochromes, which are the products of tryptophan metabolism, and the suggested biosynthetic pathway is shown in this figure. Here the various steps in biosynthesis of eye pigment in Drosophila is shown. Here tryptophan is converted into formyl kynurenine by the action of tryptophan oxidase. Formyl kynurenine is further converted into kynurenine by the action of kynurenine formamidase. Kynurenine is further converted into 3-hydroxyl kynurenine by the action of kynurenine 3-hydroxylase. 3-hydroxyl kynurenine is further converted into the pigment umochrome. The step which has been blocked can be known either by accumulation of an intermediate product or by recovery of trait due to supply of another intermediate product. This will be illustrated by two additional examples. It is important to notice that if there is a mutation in the brown pigment umochrome pathway, there will be a missing of the semi-dull effect of the brown pigment umochrome and the color of eye will be bright red. The second important point to note is that mutants were named not based on the corresponding biochemical defect, but it is named based on their actual eye color. For example, the mutant brown has a brown eye color. Now let us see about the arginine synthesis in Neurospora. In Neurospora, following three kinds of mutations for arginine synthesis were isolated. 1. Mutations which grow only when arginine is supplied and do not grow with the help of either ornithine or citrulline alone. 2. Mutations which grow only when either citrulline or arginine is supplied but do not grow when only ornithine is supplied. 3. Mutations which can grow only when either ornithine or citrulline or arginine is supplied. These mutants indicate that in the first case, 1. Mutants are incapable of utilizing ornithine or citrulline. In the second case, 2. Mutants are incapable of utilizing ornithine, but in the last case, 3. Only the precursor cannot be utilized. In this biosynthetic pathway, one set of mutant strains required arginine to grow on a minimal medium, as shown in this table. The mutations mapped into 3 different locations on separate chromosomes, even though the same supplement, arginine, satisfied the growth requirement for each mutant. The auxotropes for each of the 3 loci, arginine 1, arginine 2 and arginine 3 genes, differed in their response to the chemical compounds ornithine and citrulline, which are related to arginine. The arginine 1 mutants grew when supplied with ornithine, citrulline or arginine in addition to the minimal medium. The arginine 2 mutants grew on either arginine or citrulline but not on ornithine. The arginine 3 mutants grew only when arginine was supplied. Let us see about synthesis of tryptophan in bacteria and Neurospora. Salmonella typhimurium. In Salmonella typhimurium, 4 kinds of mutants for tryptophan synthesis could be isolated. These mutations are listed in the table and include the following. 1. Tryptophan 8 does not have the ability to synthesize anthranylic acid, but the growth can be restored by addition of either of the following compounds: anthranylic acid, indole glycerol phosphate, indole or tryptophan. 2. Tryptophan 2 and tryptophan 4 cannot grow on either the minimal medium or on the supplemented medium with anthranylic acid, but can grow on addition of either indole glycerol phosphate or indole or tryptophan. 3. Tryptophan 3 can grow only on a supplemented medium with either indole or tryptophan, but not on any other medium. 4. Tryptophan 1, 6, 7, 9, 10, 11, etc. can grow only on a supplemented medium with a tryptophan and on no other medium. These different mutants indicate interruption of biochemical pathway at different steps. Escherichia coli. In E. coli also 5 different kinds of mutations called tryptophan A, tryptophan B, tryptophan C, tryptophan D and tryptophan E were identified. These mutations are similar to those described for Salmonella and are shown in this figure and are also summarized in the table. The biosynthetic pathway shown in this figure was traced out using the mutants described above for Salmonella, Typhimurium and Escherichia coli. Tryptophan synthetase in Neurospora and bacteria. It has been shown that the last 2 steps of tryptophan metabolism in Neurospora as well as in bacteria are controlled by the same enzyme tryptophan synthetase, so that even mutants lacking the ability to utilize indole do not have tryptophan synthetase enzyme. Tryptophan synthetase is an enzyme that catalyzes the biosynthesis of tryptophan. It is typically found as an alpha to beta 2 tetramer. The alpha subunits catalyze the reversible formation of indole and glyceraldehyde 3 phosphate from indole 3 glycerol phosphate. The beta subunits catalyze the irreversible condensation of indole and serine to form tryptophan in a pyridoxal phosphate dependent reaction. In Neurospora, this enzyme consists of only one component, but in E. coli and Salmonella it has 2 components. 1. A component affecting penultimate step in the figure, so that tryptophan A mutants lacking A component can be supplemented by both indole and tryptophan, and 2. B component which affects last step in the figure, so that tryptophan B mutants lacking B component must be supplemented directly with the tryptophan. It has also been shown that in mutants for this enzyme protein is synthesized, but it is unable to perform enzymatic function. The presence of 2 components A and B of tryptophan synthetase enzyme synthesized under the control of 2 different genes suggests that a particular enzyme may consist of more than one polypeptides, each synthesized under the control of a different functional unit called cistron. Therefore, it was suggested that the concept of one gene, one enzyme proposed initially by Beadle and Totem should be modified into one cistron, one polypeptide hypothesis. Gene sequences and enzyme sequences in biosynthetic pathways. In this section, earlier we have shown that each step of a biosynthetic pathway is controlled by a specific enzyme synthesized under the influence of a specific gene. In several cases, although not universally, it has been shown that the genes involved in different steps of a biosynthetic pathway are clustered and found in the same sequence in which their corresponding enzymes are used in the biosynthetic pathway. This will be illustrated with the help of 2 examples. Tryptophan metabolism in Salmonella. It has been earlier shown that for tryptophan synthesis in Salmonella, 5 enzymes are needed for 5 steps earlier described as 1, 2, 3, 4, 5. It was found that the genes for 3 enzymes in Salmonella are found in the same order in which the steps are found in the biosynthetic pathway. Histidine metabolism in Salmonella. More than 500 mutations for histidine metabolism in Salmonella were mapped. These were linked in 9 closely linked loci A to H, each affecting a different enzyme in the synthesis of histidine. The gene sequence was found to follow largely the metabolic sequence. For several other traits including proline, threonine and isoleucine, valine metabolism in Salmonella, a similar clustering of genes was observed. This suggests that at a primitive stage, biochemical synthesis during evolution must have first organized on the chromosome itself. One gene, one enzyme hypothesis. One of the first proofs for one gene, one enzyme hypothesis came from a Arginine biosynthetic pathway. This pathway consists of 8 chemical reactions, each catalyzed by a specific enzyme, and the presence of each enzyme is controlled by a single distinct gene, as specific enzymes are absent in mutants for specific gene. One gene, one enzyme was later modified to one gene, one polypeptide hypothesis because it was found that an enzyme may be composed of more than one polypeptide chain and each polypeptide chain was synthesized from a separate gene. The relationship between genes and enzymes was first precisely emphasized by George Wells Beadle, an American scientist in the field of genetics and Nobel Prize winner in physiology or medicine. Along with Edward Laurie Totem, an American genetist who shared half of the Nobel Prize in physiology or medicine with George Wells Beadle in 1946, while working with the mutants of Neurospora crassa and proposed one gene, one enzyme hypothesis. This hypothesis states that the genes act through the production of enzymes, with each gene responsible for producing a single enzyme that in turn affects a single step in a metabolic pathway. Beadle's collaboration with Boris Frissi in the studies of the brown pigment homochrome in Drosophila led him in search for the ideal organism to use in further experiments for extending this work. Neurospora crassa, the pink bread mold, can grow on medium only with organic salts, a simple sugar and with the vitamin biotin. The growth medium for Neurospora containing only these components is called minimal medium. Beadle and Totem believed that Neurospora must be capable of synthesizing all the other essential metabolites such as the purines, pyrimidines, amino acids and other vitamins de novo. Furthermore, they believed that the biosynthesis of these growth factors must be under genetic control. If so, it would be expected to produce mutant strains with additional growth factor requirements if mutations in genes whose products are involved in the biosynthesis of essential metabolites. Beadle and Totem tested this prediction by irradiating conidia, that is asexual spores of wild type Neurospora with ultraviolet light or X-rays and screening the clones produced by the mutagenized spores for new growth factor requirements, and this is very clearly shown in this figure. In order to select strains with a mutation in only one gene, Beadle and Totem studied only mutant strains that yielded a 1:1 mutant to wild type progeny ratio when crossed with the wild type. They identified mutants that grew on the medium supplemented with all the amino acids, purines, pyrimidines and vitamins, which is called as the complete medium, but could not grow on minimal medium. They analyzed the ability of these mutants to grow on medium supplemented with just amino acids or just vitamins and so on. For instance, Beadle and Tatum identified mutant strains that grew on medium in the presence of vitamins but could not grow in the medium supplemented with amino acids or other growth factors. They next investigated the ability of these vitamin requiring strains to grow on the media supplemented with each of the vitamins separately. In this way, Beadle and Tatum demonstrated that each mutation resulted in the requirement for one growth factor. By correlating Beadle and Tatum's genetic analysis with the biochemical studies of the mutant strains, they revealed in several cases that one mutation resulted in the loss of one enzyme activity. For this work, Beadle and Tatum received a Nobel Prize in 1958 and was soon verified by similar studies of many other organisms in many laboratories. The one gene, one enzyme concept thus became a central tenet of molecular genetics. One gene, one enzyme hypothesis is a central tenet in molecular genetics and provided a basic explanation of how genes work at the molecular level. Classical genetics thought of a gene as a particle on a chromosome. To molecular genetics, however, a gene is a sequence of DNA nucleotide bases that codes for a product. Most often, the gene product is a polypeptide. Each gene specifies only polypeptide of a protein, a molecule that may contain one or more different polypeptide. It was necessary therefore to change the concept of one gene, one enzyme hypothesis to one gene, one polypeptide hypothesis. Now let us summarize the following discussion on analysis of mutations in biochemical pathways and one gene, one enzyme hypothesis. Microbial genetics is concerned with the transmission of hereditary characters in microorganisms. Microbial genetics has played an important role in developing the fields of cell and molecular biology and also in agriculture, medicine, food and also pharmaceutical industries. The genetic material is DNA in both eukaryotic and prokaryotic microbes. RNA viruses are the only exceptions. The heritable changes in the DNA, that is, mutations occur spontaneously and mutagenic agents increase the rate of mutation. The reactions involved in the biosynthesis of a compound occur in a precise sequence so that the product of previous reaction serves as substrate for the next reaction. A biosynthetic pathway can be worked out using biochemical mutations, since mutation in a particular gene leads to block in a particular step of biosynthetic pathway. It was suggested the concept of one gene, one enzyme hypothesis by Beadle and Tatum states that each gene controls a specific enzyme. One gene, one enzyme was later modified to one gene, one polypeptide hypothesis. Now let me give you certain assignments based on the discussions what we had. 1. Describe the experiment performed by Beadle and Tatum proving one gene, one enzyme hypothesis. 2. Give outlines of the biosynthesis of arginine in E. coli. How Ames test was developed to evaluate the mutagenicity of a chemical. Briefly explain the experiment. 4. Explain the steps involved in the biosynthesis of eye pigment in Drosophila. 5. Write short notes on gene sequences and enzyme sequences in biosynthetic pathways. I will also give you certain references for your further reading. 1. Book with the title Cell and Molecular Biology, authored by D. Robertis and Robertis in the year 1991 and published by Leah and Febiger, Washington. A second book is with the title Genetics and the Origin of Species, authored by Dobzhansky in the year 1961, published from Columbia University Press, New York. A third book is with the title Principles of Genetics, authored by Gardner and Nestad in the year 1984, published by John Wiley, New York. A fourth book is with the title Genes, authored by Levin in the year 1994, published by Oxford University Press, London. A fifth reference is with the title Polyploidy, authored by Lewis in the year 1980, published by Plenum Press, New York. A sixth reference is with the title Chromosome Technique, Theory and Practice, authored by Sharma and Sharma 1980, published by Aditya Books, New Delhi. 7. A book with the title Principles of Genetics, authored by Sinnett, Dunn and Dobzhansky in the year 1958, published by McGraw-Hill, New York. I will also give you certain websites for your further reference. One, www.genetics.org. Second one, www.theagricos.com. We have come to the end of this session. We will meet in another session with a new topic. Till then, bye.